Welcome. In this explainer, we are tackling a topic that is absolutely fundamental to all life: transport in cells. Now, this is a core part of the AQA GCSE Biology specification, and honestly, mastering it is key to understanding how every living thing, including you, actually works.
So just think about it for a second. Every single one of your trillions of cells is like a tiny, bustling city. It needs a constant supply of things like oxygen and nutrients, and it needs a way to get rid of all its waste products. But it's surrounded by a barrier, a membrane. So the big question is, how does anything get across? That's the problem we're going to solve today.
Okay, here's how we're going to break it down. First, we'll look at the cell membrane's job as a kind of border control. Then we'll get into the three key transport methods you absolutely have to know: diffusion, osmosis, and active transport. And to finish off, we'll pull it all together in a super handy comparison table, which is perfect for your revision.
Right, first things first, cellular border control. You know, the cell membrane isn't just some simple bag. Nope, it's an incredibly smart barrier that's constantly managing all the traffic coming in and going out of the cell. So, if you picture the cell as a tiny factory, it needs to bring in all its raw materials, stuff like oxygen and nutrients, and at the same time, it's got to ship out its waste products, like carbon dioxide and urea. It's this constant flow of traffic. To manage all that, the cell uses three key transport methods, and these are exactly what you need to nail for your exam. Let's kick things off with the most straightforward method: diffusion. This is kind of the default setting, the no energy required way for substances to move across the membrane. Just think of it as particles naturally spreading themselves out. And here it is, the official AQA definition you'll want to remember. The most crucial part of this is the phrase net movement. See, particles are always moving around randomly, but the net movement is always from where there's a lot of them to where there's fewer of them. They move down a concentration gradient. Now, diffusion doesn't always happen at the same speed, and you can bet the exam board loves asking about what affects its rate. There are three big ones. First, the concentration gradient. The steeper it is, so the bigger the difference in concentration, the faster diffusion happens. Second, temperature. Higher temperatures need more kinetic energy, so particles move around faster and spread out quicker. And finally, surface area. A larger surface area just gives more space for particles to cross, which, you guessed it, speeds things up.
And that last point brings us to a massive, crucial concept for your exams: the surface area to volume ratio. Honestly, this idea is absolutely key to understanding why cells are so tiny and why big organisms like us need complex transport systems. You see, a tiny single-celled organism has a massive surface area compared to its volume, so simple diffusion is totally fine for it. But for a big organism like a human, our surface area to volume ratio is tiny. There's just no way diffusion alone could get enough oxygen to all our cells deep inside our bodies. That's exactly why we have things like lungs and intestines. They are specialized exchange surfaces, brilliantly designed to maximize that surface area. Okay, on to our next process: osmosis. Now, a great way to think about this is as a special case of diffusion. It is diffusion, but with two very important rules. It's only for water, and it only happens across a partially permeable membrane. That's a membrane with tiny little holes that let small molecules like water through, but block bigger ones. And here's the precise definition. The trick is to think about the concentration of water. A dilute solution, like pure water, has a really high concentration of water molecules. But a concentrated solution, say sugary water, has fewer free water molecules because the sugar is in the way. So, water naturally moves from where it's more concentrated to where it's less concentrated. In other words, from the dilute to the concentrated solution. And guess what? This is the science behind one of your required practicals. You know, the one with the potato cylinders? It's osmosis that makes them gain or lose mass in different solutions. The examiners will definitely expect you to know this experiment inside out. So you remember these steps, right? You cut up the potato, weigh it, and pop the pieces into different solutions, from pure water all the way to a really concentrated sugar solution. And what you're seeing is osmosis in action. In pure water, the potato gains mass because water moves in. In the concentrated solution, it loses mass because water moves out. It is a perfect way to demonstrate the principle. So, we've covered the passive, go-with-the-flow methods. But what happens when a cell needs to move something against the flow? Well, for that, it needs active transport. Think of this as the complete opposite of diffusion. It's like trying to push a boulder uphill, and that, of course, is going to require some energy.
This definition tells you the two most important things you need to know. First, the movement is against a concentration gradient. It goes from low to high. And second, this is crucial, it requires energy. So where does that energy come from? It comes from respiration. No respiration, no active transport. Simple as that.
So what does this actually look like in real life? Well, these are the two classic examples you've got to know. First up, in plants. Root hairs need to pull in mineral ions from the soil, even when the concentration of those minerals is already way higher inside the root. That's active transport. Second, in us. Our small intestine absorbs sugar molecules from our food into our blood, even when our blood already has plenty of sugar. Both are totally vital and both work using active transport.
Okay, we've covered the three key processes. Now for what might be the most important part of your revision, putting them all side by side to really get the differences clear in your head. This is where the marks are really won or lost in an exam.
Right, this table is your golden ticket for revision. Let's just walk through it. Look at the direction and energy columns. You can see diffusion and osmosis both move stuff from high to low concentration, and neither needs any energy. They're passive, they just happen. But active transport, it's the total opposite. It moves substances from low to high against the gradient, and it absolutely requires energy. And then look at what's actually being moved. Diffusion is for solutes and gases, but osmosis, it's a special pass for water only. If you can memorize this table, you'll have these processes completely locked down.
So, let's leave you with one final thought to really connect all the dots. We've just established that active transport is essential and that it's powered by respiration. So, what do you think happens to a cell when it can't respire properly? What essential processes would just grind to a halt? Thinking about these links is the real key to mastering biology.